In today's lesson, we're going to discuss what is cellular respiration. After you hear a word respiration, you may think about breathing. During breathing, the oxygen is entered with each inhale and carbon dioxide is released with each exhale. You may also notice that breathing is a part of physiological respiration and functions because it brings oxygen into the lungs and expels carbon dioxide. Cellular respiration is a chemical process by which energy is obtained within the individual cells from the biomolecules. All living organisms, from a single cell to the most enormous creature all over the world, undergoes the respiration. Cellular respiration requires oxygen, which occurs in most organisms. It also provides a supply of usable energy for the cells in the form of ATP. On the screen you can see the reaction, respiration reaction. Glucose react with oxygen to give you carbon dioxide, water and energy. Glucose is undergoing oxidation because it loses an hydrogen atom, and oxygen is going under the reduction process because it gains a hydrogen atoms to form water, while glucose form the carbon dioxide. Again, what we said about respiration is a metabolic process consisting of a series of steps to convert chemical energy such as glucose or sugar into a usable form of energy like ATP in the cell. Reaction involved in cellular respirations are catabolic reaction, which means that it will break down large organic molecules into the smaller form. Respiration is a basis of life that occurs in all living forms. In most multicellular organisms, cellular respiration occurs in the form of aerobic respiration. Process of respiration involves the breakdown of high energy bond which releases energy in form of the ATP. It is a combustion reaction, but the processes in the cell occurs in a slow, controlled manner to release energy via the series of reaction. Most of the reaction in cellular respiration are the redox reactions in the presence of the strong oxidizing agents like molecular oxygen. Chemical energy produced during the cellular respiration is stored in form of ATP, which releases energy by the breaking of bond of the third phosphate group during processes like biosynthesis, locomotion and active transport of the molecule. Different biomolecules and structures are involved during the process of respiration. A different set of enzymes catalyzes different steps of cellular respiration, and all of which are found within the cell. Let's look at the biomolecules. Number one is ATP. ATP, adenosine triphosphate, is an inorganic compound that acts as an energy carrying molecule by capturing energy produced from the chemical reaction. ATP is a nucleotide which consists of three main structural units like nitrogenous bases, adenine, sugar unit, ribose, and three phosphate groups bound to the ribose backbone. ATP doesn't act as a storage unit. ATP doesn't act as a storage unit for energy like carbohydrates and proteins, but act as a shuttle to release energy during the energy consuming activities. The release of energy by ATP occurs as a result of breaking down of phosphate bonds to form ADP or AMP molecules. Most of the ATP in the cell is synthesized in mitochondria as it is considered the powerhouse of the cell, while some ATP might be produced in the cytoplasm. Next molecule is NAD. NAD is your nicotinamide adenine diphosphate. NAD is a coenzyme that plays a central role in cellular respiration as it acts as a mean of electron transport. The molecule consists of two nucleotide units where one contains adenine as the nucleobase and the other contains the nicotinamide unit. Besides, two phosphate groups are attached to the nucleotide unit as you can see in the diagram. NAD exists in the two different forms based on its oxidation state. NAD plus is oxidized state and NADH is the reduced state. NAD is involved in redox reaction where it becomes reduced by accepting electrons and oxidizes by donating those electrons to the other molecule. It is synthesized in the body from the smaller unit of amino acid like the tryptophan and aspartic acid. Next is flavin adenine dinucleotide FAD. FAD is metabolic coenzyme that is involved in various enzymatic reactions in the body as an electron carrier. It has a similar structure to NAD with two nucleotide unit where one consists of adenine as the nitrogen base and whereas the other consists of the flavin unit. FAD is synthesized in the body from the riboflavin and two molecules of ATP. The phosphorylation of riboflavin by ATP will result in the formation of FMN. The transfer of an AMP unit from the ATP then results in the formation of FAD. In the case of cellular respiration, the FAD involved exists in two oxidation states, FADH and FADH2. Due to their multiple oxidation states, FAD molecules are involved in the transfer of electron from one molecule to another. Then we have the location of cellular respiration. Cellular respiration takes place in the individual cell to produce energy for the cell. Within the eukaryotic cell, cellular respiration begins in the cytoplasm. The first step for cellular respiration is glycolysis that takes place in the cytoplasm as the enzymes required for glycolysis are present in the cytoplasm. The end result of glycolysis steps are transported into mitochondria of the cell where the rest of the steps of cellular respiration take place. The next step, like the citric acid cycle, oxidative phosphorylation, occurs in the outer and inner membrane of the mitochondria. In the case of prokaryotic cell, however, the steps of cellular respiration occurs in the cytoplasm as they do not have defined cell organelle. So, in the cytoplasm, glycolysis will occur. Other steps like citric acid oxidative phosphorylation will occur in the inner and outer membrane of mitochondria. Three steps of cellular respiration are: number 1, glycolysis, where glucose will change into pyruvate, then citric acid cycle, and oxidative phosphorylation is the third step. The steps and reaction involved in cellular respiration might be different in different types of cellular respirations. One of the type is aerobic respiration. In aerobic respiration, one glucose molecule will combine with an oxygen molecule and ADP to form carbon dioxide, water and energy. Aerobic respiration is the most efficient pathway of cellular respiration that produces the largest number of ATP. 36 ATP is produced in this process. Then we have anaerobic respiration. Anaerobic respiration equation involved depend on the pathway utilized. Anaerobic pathway are less efficient than aerobic respiration as they produce a lesser number of ATPs. In alcoholic fermentation, one molecule of glucose degrades into ethyl alcohol, carbon dioxide and energy. The process takes place in absence of oxygen. And in lactic acid fermentation, both of these are type of anaerobic respiration. Lactic acid fermentation, one molecule of glucose is degraded into the lactic acid and energy. This respiration also occurs in the absence of oxygen. The first step involved in the cellular respiration is glycolysis. Glycolysis is where the glucose molecule is catabolized to form a pyruvate through a series of 10 steps. You can see in the figure the 10 steps by which glucose is catabolized to form the pyruvate. Glycolysis is the initial step of glucose metabolism, which is the common pathway in both aerobic and anaerobic respiration. Overall sequence of reaction in glycolysis might differ from one species to another in its regulation and fate of pyruvate. During glycolysis, the 6-carbon compound like glucose break down into the 3-carbon compound like pyruvate with release of 2 molecules of ATP. In aerobic respiration, glycolysis is prelude to the citric acid cycle and electron transport chain, which are responsible for the production of most of the ATPs. The product of glycolysis can proceed in one of the three different pathways depending on the availability of oxygen and metabolic activities. Let's look at the steps involved in glucose glycolysis. Number one is glucose. One of the phosphate molecule will release to form ADP and it will become glucose 6-phosphate. Next step is fructose 6-phosphate, then fructose 1,6 bisphosphate, which will give you dihydroxyacetone phosphate and glycerol aldehyde 3-phosphate. Glycerol aldehyde 3-phosphate, two molecules will give you 1,3 bisphosphoglycerate, two molecules, then 3-phosphoglycerate, 2-phosphoglycerate, and the phosphoenol pyruvate, and finally pyruvate. You only have to know the names of these steps, no need to go in details. The next step is pyruvate oxidation. Oxidation of pyruvate is the second step of aerobic respiration occurring, representing one of the three possible fates of pyruvate molecule. Pyruvate molecules are the end product of glycolysis, which is the common pathway in both aerobic and anaerobic respiration. Fate of pyruvate is determined by the availability of oxygen and metabolic conditions. Oxidation of pyruvate occurs in the presence of oxygen after pyruvate molecules are moved to the mitochondria from the cytoplasm. The pyruvate derived from the glycolysis is dehydrogenated to yield acetyl coenzyme A and carbon dioxide by the enzyme pyruvate dehydrogenase complex. The enzyme is found in mitochondrial matrix of eukaryotes and cytoplasm of the prokaryotes. Pyruvate oxidation acts as a link between glycolysis and citric acid cycle in the case of aerobic respiration. During pyruvate oxidation, a total of 3 ATPs are formed after the entry of NADH to the electron transport chain. This reaction represents the pyruvate oxidation where pyruvate coenzyme A will react with NAD to form acetyl coenzyme A and NADH. Citric acid cycle, or also known as Krebs cycle, is the process of complete oxidation of acetyl coenzyme A to release carbon dioxide and water molecule. Most universal pathway of aerobic metabolism of energy-rich molecule is the citric acid cycle. Reaction of the cycle provides electron to electron transport chain which reduces oxygen while generating ATP. This cycle is important not only for carbohydrate metabolism but also for other biomolecules like amino acids and fatty acids. The cycle can occur in presence of oxygen as energy-rich molecule like NADH, NAD plus and FAD can retrieve ATP from their reduced form by transfer of electron to the molecular oxygen. There are two main purposes of citric acid cycle, which include disposition of carbon and hydrogen atoms and the conversion of potential chemical energy into the metabolic energy in form of ATP. A total of 12 ATPs are formed during the complete oxidation of a single molecule of acetyl coenzyme A. Out of 12 ATP molecules, only one ATP molecule is produced directly from the cycle. The rest are generated after the entry of high energy molecule into the electron transport chain. In the citric acid cycle, step number 1 is a 2-carbon acetyl group carried by coenzyme A is transferred by oxidoacetate forming a citrate. Citrate will rearranged into its isomer to form isocitrate. Then isocitrate is oxidized to alpha ketoglutarate One carbon is removed and released as a carbon dioxide, and NAD plus is reduced to NADH and hydrogen. Alpha-ketoglutarate is oxidized into succinyl coenzyme A. One carbon is removed and released as carbon dioxide, and NADH is reduced to the NADH plus hydrogen. The release of coenzyme A from succinyl coenzyme produces succinate. The energy released converts GDP to GTP, which is in turn converts ADP to ATP by substrate level phosphorylation. This is the only ATP made directly in the citric acid cycle. Succinate is oxidized to fumarate. The 2 electron and 2 proton removed from succinate are transferred to FAD, producing FADH2. Then fumarate is converted into malate by the addition of water molecule. Malate is oxidized to oxaloacetate, reducing NAD plus to NADH and hydrogen. Oxaloacetate can react with acetyl coenzyme A to re-enter the citric acid cycle. To remember all the citric acid cycles compound, you can use the mnemonic O. Can I keep some succinate for myself? O stand for oxaloacetate, C for citrate, I for isocitrate, K for ketoglutarate, then S for succinyl coenzyme A, another S for succinate, F for fumarate, and M for malate. What is citric acid cycle? It is a series of chemical reaction that finishes the breakdown of glucose. 3-carbon pyruvate from the glycolysis breakdown to 3 carbon dioxide. Where does it occur? It occurs in the mitochondrial matrix. Pyruvate from cytoplasm get transferred across the inner mitochondrial membrane into the matrix. And where does the released energy end up? As the high energy electron in the electron carrier NADH and FADH, and electron pass on to the electron transport chain. Next step is oxidative phosphorylation. Oxidative phosphorylation or electron transport chain is the final step of aerobic respiration. It consists of chain of redox reaction to synthesize ATP molecule. During the process, electrons generated in citric acid cycle are transferred from the organic compound to oxygen while simultaneously releasing energy in form of ATP. The transport of electrons occur between 4 large protein complexes that are present in the inner mitochondrial membrane. The chain consists of a series of protein with tightly bound prosthetic groups that are capable of accepting and donating electrons by virtue of their multiple oxidation state. The number of ATP synthesized during the oxidative phosphorylation depends on the energy-rich molecule passing from the electron. For example, NADH produces 3 moles of ATP, whereas FADH produces 2 moles of ATP. Oxidative phosphorylation is essential for metabolism of all biomolecules as all the metabolic reactions converge at this stage. There are different chemical groups that act as electron carrier during the transport of electron through the chain. Besides, there are 4 important coenzyme complexes that catalyzes the transfer. Then we have lactic acid fermentation. Lactic acid fermentation is type of fermentation. It is a form of anaerobic respiration in which complex organic compound like glucose are converted into lactic acid while releasing some amount of cellular energy. Lactic acid fermentation begins with glycolysis, which breaks down a single molecule of glucose into 2 moles of pyruvate in the presence of oxygen. The pyruvate formed undergoes pyruvate oxidation to form carbon dioxide, but in the absence of oxygen or in the presence of limited oxygen, the pyruvate formed undergoes different form of fermentation. Lactic acid fermentation occurs in some of the anaerobic organisms that live in the low oxygen environments or in muscle cells during the exercise. The enzyme involved in lactic acid fermentation is lactic dehydrogenase, which catalyzes the conversion of pyruvate into lactic acid along with the oxidation of NADH into NAD+. NAD+ formed in lactic acid fermentation produces two ATPs through the electron transport chain. The lactic acid produced during lactic acid fermentation in muscle cell is accumulated in the muscle. The lactic acid is then passed to the liver where it is converted back to the pyruvate so that it can be utilized during the aerobic respiration. Besides being a pathway of metabolism, lactic acid fermentation is also considered a desirable method of processing and preserving vegetables because the method is of low cost and yields high acceptable and diversified flavor. Lactic acid fermenting bacteria are industrially important as they are involved in production of yogurt, cheese and other dairy products. The next is alcoholic fermentation, also anaerobic respiration. This is the most common pathway of anaerobic respiration is alcoholic fermentation in which the carbohydrate molecule is partially oxidized to form alcohol as a byproduct. The conversion of pyruvate to ethyl alcohol in alcoholic fermentation is a two-step process where one is an oxidation reaction and the other one is redox reaction or reduction reaction. So this is the redox reaction. The initial step of alcoholic fermentation is the same in both aerobic respiration and lactic acid fermentation. The substrate in alcoholic fermentation, like in lactic acid fermentation, is pyruvate. In the first step, the pyruvate molecule produced from glycolysis is decarboxylated by the catalytic action of pyruvate decarboxylase to produce acetylaldehyde. Acetylaldehyde is then reduced to ethanol by NADH in the presence of an alcohol dehydrogenase enzyme. NADH is oxidized to form NAD+, which produces ATP via an electron transport chain. The end products of alcoholic fermentation are ethanol and carbon dioxide. Production of alcohol from the carbohydrate is industrially important to produce alcoholic drinks like beer and wine. Alcoholic fermentation is the mode of cellular respiration in yeast and other microorganisms. However, the accumulation of large quantities of alcohol might be harmful to some of the microorganisms. This diagram represents a summary of lactate fermentation and alcoholic fermentation. The fermentation which occurs in the cytosols convert NADH to NAD+, allowing the electron carrier to cycle back to glycolysis. This process keeps glycolysis running, which continued with continued production of the ATP. Then we have methanogenesis. Methanogenesis is a unique form of anaerobic respiration that involves the breakdown of carbohydrate molecule into methane carbon dioxide as a byproduct. Most organisms that respire via methanogenesis belong to the domain Archaea that live in close association with anaerobic bacteria. Methanogenesis is an essential metabolic process in such organisms as it is the final step of biomass decomposition. It is an anaerobic process and the terminal electron acceptor in methanogenesis is a carbon compound. Common electron acceptor in methanogenesis are acetic acid or carbon dioxide. In addition to helping in breakdown of large complex organic molecules, methanogenesis is also important for the decay of organic matter. During advanced stage of biodecomposition, all other electron acceptors except carbon dioxide become depleted. In such cases, remaining organic matter is degraded by methanogenesis where CO2 or carbon dioxide act as the electron acceptor. It is also performed by some symbiotic bacteria that exist in the digestive tract of ruminants. This enables the digestion of organic matters which otherwise wouldn't be degraded by the cattle. Let's look at the products of cellular respiration. Number one is ATP. Most important product of cellular respiration is ATP or energy. ATPs are the carrier molecules that store energy in form of phosphate bonds which are then released after breaking of those bonds when required. ATP is converted into ADP after the release of energy. The ADP molecules then gain energy to form ATP molecules. ATP is converted into ADP after the release of energy. The ADP molecules then gain energy to form the ATP molecule. The ability of the molecule to continuously break and make phosphate bonds allow a single ATP molecule to be used multiple times to transport energy from one place to another. Efficiency of cellular respiration is determined by the number of ATP molecules produced at the end of the process. In aerobic respiration, a total of 36 ATPs are formed at the end of the process. The number is quite low in case of anaerobic respiration. The other product is carbon dioxide. Carbon dioxide is a universal product of all cellular respiration. It is considered a waste product and thus is removed out of the cell by some means. Presence of large quantities of carbon dioxide in the cell might induce toxicity as it increases the pH of the cytoplasm. In the presence of water, carbon dioxide might form carbonic acid, which might be toxic to the some cells. The release of carbon dioxide from the cell is mostly an active process and requires some amount of energy. Besides ATP and carbon dioxide, other products are also formed during the cellular respiration, which depends on the type of cellular respiration. In the case of fermentation or anaerobic respiration, common products include acetic acid, ethyl alcohol, methane, lactic acid, and propionic acid, etc. Water molecules are also formed during aerobic respiration after the capture of electron by the oxygen molecule. Lastly, the purpose of cellular respiration. Most important purpose of cellular respiration is the production of energy required for the different processes in the cell. The end intermediate product of cellular respiration can be used for biosynthesis of various biomolecules in the body. Cellular respiration is also an essential part of carbon cycle which work as a natural waste management system. Steps involved in cellular respiration are also important for the metabolism of other biomolecules like amino acid and fatty acid. Besides, other products of cellular respiration have different industrial application. Anaerobic respiration processes like alcoholic fermentation and lactic acid fermentation are industrially important to produce various commercial products. This brings us to the end of cellular respiration.